So dear students, once again, today we will talk about the topic of DNA replication. So without wasting your time, we will start the topic. So the heading is the DNA replication. You will focus on this red marker, then I will explain with word by word. First of all, we would have the simple definition. What do you mean by the word of replication? To double the something, to double the amount of something. For example, you want to give 50 rupees to two people and you want that each person will get 50 rupees. Then definitely you would have to add 50 rupees once again. Similarly, replication means to double the amount of DNA. But why? Why it is necessary? So the answer is very simple. For the equal distribution of the DNA among the new cells. So it was the first point. Answer is distribution. Next, there is an MCQ. When replication actually occurs, G1 phase, G2 phase, S phase or G0 phase. Actually, replication occurs in the S phase of the cell cycle. It means the amount of replication, the amount of DNA is doubled during replication only when cell divides. And when cell is non-dividing stage, then DNA would not be replicated. First of all, the second main outline is the origin of the replication, means from where it is started. According to the given syllabus of F.sc and the other books which are taught here, in all the books, the replication has been given about the prokaryotes. And definitely, we will also talk about the prokaryotic replication. And now, we would have the idea of origin. What is the origin? First of all, we will talk about the origin of replication in bacteria. This is the bacterial circular chromosome. Keep remember, in prokaryotes like bacteria, the replication is started only from the single origin. And this single region contain the rich amount of adenine and thymine. What I am saying actually, this is the whole genome or the circular chromosome of the bacteria. And at this whole chromosome or the circular DNA in the bacteria, the replication will be started only from the single location, only from the single point. And that single point contain a rich amount of adenine and thymine. But in the case of eukaryotes, like these are the chromosomes, this is a simple chromosome. And definitely everybody knows that it's also made up of DNA. In eukaryotic chromosome, the replication has many origin, means it can be started from the multiple points and we cannot precise that which type of nucleotides are found in the eukaryotic DNA from where the replication is started, because it's vary among point to point, among location to location. Have you noticed something? There are two types of chromosome have been given. This is a single chromatid chromosome, while this is the double chromatid chromosome or the two chromatid chromosome. What is the difference among them? The main difference is the replication. The main difference is the replication. It is called the post-replicative chromosome. So, students, we were talking about these type of two chromosome. The chromosome with single chromatid means a chromosome without replication, while the chromosome with true chromatid means the replicated chromosomes. Now you can link this point with this one, this MCQ. You can also say this is the post S phase chromosome. Both MCQs have been given many times in MDCAT session, and the one confusion I want to clear to all of you, most of the people think that the molecular biology has not been given in the syllabus of MDCAT, but keep remember, sometime the questions are constructed from the DNA, the molecular biology, the chapter which is lie in the second number of the first year. Well, replication has been divided into three main phases. The first phase is initiation, means the beginning of something. These events will be completed among the replication process. The first one is unwinding. Unwinding of DNA. Look upon here. This is the double-stranded DNA. And here the replication is still not started. Okay, now the replication will be started. How? During the initiation, the replication bubble is formed. Replication bubble. What do you mean by this bubble? This is the bubble. Have you noticed something? There was the specific type of sequence, specific type of region from where the replication was started. And how it was started? Keep focus. These two strands become separated from each other. Means the hydrogen bonds which were present among them, it had been broken. That's why this bubble-like appearance will be appeared. Now, second important point. Most of the people are confused among the bubble and the term of replication fork. This whole structure is called bubble and the each side of the bubble is called fork. Do you know this is the MCQ that each bubble means it has two forks. The replicating fork. The first one and the second one means each bubble is consist of two replication forks. That's all. And how the bubble was formed, already told you, the hydrogen bond was broken down which was present between these two strands. How it was broken down? Look upon here now. Unwinding means the breaking or the separation of the DNA two strands. I am repeating, unwinding means the separation of the two strands. How the separation was completed? Separation is completed with the help of an enzyme which is called DNA helicase. This is the DNA helicase. This is the DNA helicase. Actually, it came here and it's broken down the... So students, we were talking about the concept of DNA helicase. Actually, what it's done, it's broken down the hydrogen bonds which was present between the two strands. Do you know the MCQ is given that which type of bonding is broken down by the helicase? This is the MCQ. Give the answer or remember the answer, and that's the hydrogen bond. And many times it has been given in the separate competitive exams. Listen, if we are doing the replication in vitro, means outside of the body. In vitro, these bonds are not broken down by the help of helicase. In vitro, outside of the body or in the laboratory, these bonds are broken down with the help of temperature or with the help of heat. Well, move to the next. Once the strands are become separated. Now look upon here. We are just mentioning the one side of the bubble. Means we are just talking about a single replication fork, not about the both. In each replication fork, when two strands become separated, now it will be look like that. It will be look like this. This whole structure is called replication fork. Each replication fork. Once the strands become separated, these strands must be separated from each other or apart from each other. But how it's possible? There are special types of protein and they are called single-stranded binding proteins. They are the single-stranded binding proteins. They are performing two functions here. The first one, they try to keep separate the both strands, and the second function, they protect the single strand from being degraded by the degrading enzymes, because all this process is occurring inside of the nucleus, inside of the cell in the case of bacteria. So single strands sometime become digested by some other digestive enzyme. So these single stranded binding protein providing help to the each strand. So there are the two functions. Well, now there is a special concept, action of gyrase or topoisomerase. Almost in all the books this idea has also been given. Actually, when helicase come, look upon this thread. When helicase come, what it's done, it broken down the bond hydrogen bonds between the strands. This is the helicase activity. But look upon here, when the strands become separated, sometime the coils become much more harder, like at this end. Now it is not furthering unwinding, it's not further unwinding even from this side. So this is the function of the topoisomerase. Now what's the topoisomerase does? Topoisomerase will cut this strand, and when this coil will be released, this topoisomerase will rejoin the DNA strand. Again, if you want to hear that, I will repeat that. When helicase break the bonds, it unwinds the DNA, but at some point their winding become too much coiled, and then the DNA would be unable to open it further. Then the topoisomerase comes here and it cuts the DNA from the two sides and again rejoin the DNA strands, and it will release that supercoils. This is the function of the topoisomerase. Hope you would have understand it. Okay. Next, all these properties has been done in the process of initiation, means the formation of replication bubble and the formation of the replication fork, and we just also talked about the action of helicase and single stranded binding protein. Finally, now we have reached at the extension. Polymerization means to add the nucleotides. Look upon here. Polymerization is done by the enzyme which is called DNA polymerase. This is an enzyme and it has three types: polymerase 1, polymerase 2 and polymerase 3. And all these points have been given for the clearance of your MCQs. Keep remember, among all the three, the polymerase 3 is the main enzyme because it performs the main activity of the replication. Number 2, it also performs the proofreading. What do you mean by the proofreading? During replication, if a wrong nucleotide or mistakenly a wrong nucleotide is added, it will remove that wrong nucleotide. This is called proofreading. Polymerase 2, its only function during the general life of the cell and the general damaging occurs in the cell. So it repairs the DNA, means directly it does not involve in the process of replication. Yes, polymerase 1 directly involved in the process of replication, but it will be discussed in the case of in the process of termination, which will be the last step of the replication. So what is polymerase 3? Polymerase 3 is a quite complex enzyme, is a multi subunit enzyme, means it consists on many units: alpha, gamma, epsilon, torque, but these ideas have not been given in our syllabus, so you should not need to be bothered about that. But there is a problem. DNA polymerase 3 cannot initiate the process. So how it will start the process? There are some requirements. The first requirement is the presence of primer, and the second requirement of the polymerase 3, it requires the 3 end where the hydroxyl group is found. I will explain the both. What is primer? Primer is the short strand of RNA based nucleotide. How many shorters? There are only 8 to 10 nucleotides which consist on RNA based nucleotides. Do you know the difference among the DNA based nucleotides and RNA based nucleotides? RNA based nucleotides means they contain ribose instead of deoxyribose, and they contain the uracil instead of the thiamine. Okay, this is the primer, means a short strand of RNA based nucleotide. If this is the primer, it is the 5 end of the primer, and it is the 3 end of the primer. And all of you know 5 end means the presence of phosphate group, and 3 end means the presence of hydroxyl group. Have you noticed something? We have completed both the requirements, the primer and as well the requirement of the hydroxyl, which must be present at the 3 end of that primer. Now this DNA polymerase 3 can initiate the process. Second important thing about the DNA polymerase 3, which most of the students do not about that. DNA polymerase 3 is a dimer, means it consists on 2 identical units. One is here and the second is here, and both these units are joined by a single polypeptide. There is a specific name, and in Greek language this polypeptide is presented with the word of torque. Well, you have no need to mention the idea of this polypeptide, but you must keep remember that these 2 units are joined to each other with some kind of factors, and one of them is working on this strand and the second one is working on this strand. So now finally these polymerase 3 will start to add the nucleotides. Let's suppose if there is an adenine, it will add the thiamine. If there is a thiamine, it will add the adenine. Means the complementary nucleotides will be added here, and all these nucleotides will be added with the help of this DNA polymerase 3. Means now it has been reached here from here to here. Similarly, the another unit is also present here. It will also start to add the nucleotides. So I am adding here. Have you noticed something? On this replication fork, the above replication occurred in this direction, and thus this replication occurred in the opposite direction. Technically, we will explain. This replication was happening towards the replication fork, and this replication was occurring away from the replication fork. Now students, there are 2 terms. That replication which move towards the replication fork, that newly formed strand is called leading strand, and while that strand which was made by the away from the replication fork, that is called lagging strand. And now here we have another interesting MCQ. What is the direction of the leading strand? Keep remember, the direction of the leading strand is always 5 to 3. And there is no confusion. The lagging strands also contains the 5 to 3 direction. So students, it was the part 1 of this replication process. In our part 2, we will take the start that how these directions are determined about leading and lagging strand. Hopefully you will understand and you will watch this video and try to keep watching and subscribe the channel. Thank you so much.